So by way of overview, I'm going to talk about hepatitis C as an inflammatory disease. We'll talk with an emphasis for the talk will be on chemokines, in particular CXCL10. I'll briefly introduce how HCV infection is sensed by pathogen recognition receptors. And the overarching question in my talk is going to be, how does hepatocellular sensing of HCV infection lead to CXCL10 induction? And we'll talk about the role of RIG-I and TLR3 pattern recognition receptors and type 1 and type 3 interferons in CXCL10 induction. And I hope by the end of my talk that I will convince you that in hepatocytes, CXCL10 induction does not require any interferon, and that in the context of the liver, non-parenchymal cells produce interferon that amplify intrinsic CXCL10 production from hepatocytes. So everything I'm talking about today is basically comprises the large portion of this young person here, Jessica Brownell. She seems to be everywhere in the lab at many times, many places, and she does a lot of things. And she's actually defending at the end of this month. So if you don't believe me, then maybe you should, talk to Jess. So hepatitis C is, as we all know, affects 170 million people globally. It's the leading cause of liver transplantations, and it's caused by hepatitis C virus, which is a positive sense single-stranded RNA virus in the Flaviviridae and the Hepacvirus genus. It persists in most people, and it's histologically defined by chronic inflammation and immune cell infiltration of the liver. And these cells are recruited to the liver and the sites of infection by chemokines. And in particular, CXCL10 is a major player in this immune cell and non-parenchymal cell recruitment. CXCL10 binds to and recruits type 1 immune effector cells, including or expressing the CXCR3 receptor. And these immune cells include NK cells, cytotoxic T cells, and T helper cells. And importantly, CXCL10 is induced in pretty much any model of HCV infection, and this includes cell culture, it includes chimpanzee studies, it includes the small animal models, as well as humans. Furthermore, CXCR3 positive immune cells are found in the liver of patients with chronic hepatitis C. And clinical significance is further strengthened by the observation that both serum and intrahepatic CXCL10 levels are negatively correlated with sustained virologic response to antiviral therapy. So the general pattern of how we think CXCL10 and other pro-inflammatory chemokines initiate and continue inflammation is as shown in this slide. Upon infection of the hepatocyte, it's sensed by the cytoplasmic RIG-like receptor, RIG-I, and endosomal TLR3, which signal through a couple of adapters, MAVs and TRIF, to activate the various transcription factors. And I will be talking later about the role of NF-kappa B and IRFs, or interferon regulatory factors. And the activation or engagement of RIG-I and TLR3 leads to transcriptional activation of the CXCL10 promoter, production of CXCL10. And this recruits cells from the periphery and also cells within the liver to the site of infection. And these cells, these non-parenchymal cells include CD4 and CD8 cells, NK cells, Kupffer cells, and hepatic stellate cells. These cells, upon receipt of other signals in this microenvironment, produce type 1 and type 3 interferons and other pro-inflammatory cytokines. And in about 15 to 20% of cases, this very potent response that's initiated by CXCL10 leads to resolution. But in the majority of patients, the virus infection persists, resulting sort of this continuing cycle of inflammation and recruitment. And then there are these other things that happen, deposition of collagen, as we heard about in one of the talks today, as well as changes in hepatic stellate cells. They become activated, they proliferate, and this leads to the liver disease that we see histologically. It includes fibrosis, cirrhosis, and hepatocellular carcinoma. So we really wanted to explore in detail how CXCL10 is produced during HCV infection. And so we want to figure out how to get from here to here. And the possible ways are looking at, so we're focusing on RIG-I and TLR3 because these are known pathogen recognition receptors that sense HCV infection. The other way that, and so after signaling through MABs and TRIF, you can get interferon production, and this includes type 1 interferons, alpha and beta, and type 3 interferons, or the IL-28AB and or interleukin-29 families. And these interferons signal through the JAK-STAT pathway that could potentially induce CXCL10 production. But the other way that it can happen can be sort of in an interferon-independent way, where there's direct activation of things like interferon regulatory factors, which don't require signaling through JAK-STAT. And these can directly bypass this interferon pathway to induce the chemokine. So we're going to go through these steps, and we're going to focus first on RIG-I and TLR3. So to do that, we were using a cell culture-based system, and we're using the Huh7, the human hepatoma model. And it's well known that the parental Huh7 cells, they don't express TLR3, so they will not signal through TLR3 unless you express it. But they have a functional RIG-I molecule in this. So these cells are effectively TLR3 negative, RIG-I positive. Ray Sumter and Mike Gale's lab several years ago identified a derivative of Huh7 cells called Huh7.5 cells that had a mutation in the CAR domain of RIG-I, rendering it that although the protein was expressed, it was not able to signal. So these Huh7.5 cells are basically double negative for RIG-I and TLR3. And through collaborators, we obtained an Huh7.5 cell line overexpressing TLR3 in the endosome. So these are TLR3 positive, RIG-I negative. And then we also obtained an Huh7 cell line overexpressing TLR3. So these cells are effectively double positive for TLR3 and RIG. So these are the 4 cell lines with all the possible combinations of the 2 known sensors of HCV that we're going to use, that we were using in our studies. So the first thing we did is confirmed expression and the functionality of these receptors. So on the bottom here is the TLR3 RIG status from double negative to double positive. And you can see that TLR3 is expressed appropriately where it should be. And on the right, you can see that RIG-I is expressed. And as I mentioned, in the mutant RIG cell lines, the protein is expressed, but it's non-functional. So that's why you see RIG here, RIG-I in pretty much every lane. And Sendai virus is a known trigger of RIG-I, so we also infected these cells with RIG-I, and we infected the cells with Sendai virus. And you can see that RIG-I was actually induced in some cases with Sendai virus, and that's also known because RIG-I is an interferon-stimulated gene. And using these cell lines, we then treated them with various triggers of the TLR3 or RIG-I pathways. And we simply added the synthetic double-stranded RNA poly-IC, which triggers TLR3 signaling. Or we infected cells with Sendai virus, which is a known trigger of RIG-I. And you can see on the left here is the CXCL10 mRNA production. On the right is the protein. And you can see that the cell lines responded appropriately. So the double negative cells don't really respond to either a TLR3 or RIG trigger. The RIG-I positive cell line responds very nicely to Sendai virus, as does the double positive cell. And that the TLR3 positive cell has a decent induction of CXCL10 in response to poly-IC. And you see the same thing on the protein. Again, the RIG-I cell line responds to Sendai virus, in both cases, RIG here, RIG there. And treatment with poly-IC induces a large amount of CXCL10 protein. So we were pretty confident that the cell lines that we had were responding to the appropriate triggers, and so we felt that we could proceed with HCV infection. And these are the results of a low multiplicity of infection with JFH1. On the left, we quantitated the amount of JFH1 genomes per and a defined amount of total cellular RNA. because the cell lines express different amounts of receptors. So the more receptors you have, the less permissive you are to virus infection. And you can see that cells that express both TLR3 and RIG-I are quite non-permissive to JFH1 infection. And then the Huh7.5 cells, which have neither TLR3 or RIG, have very high levels. But the bottom line is that we can get infection in all of these cell lines. And when you look at the CXCL10 mRNA response, you can see that the double positive cells express the highest levels of CXCL10 mRNA. And this also correlates at the protein level. Here, the double positive cells have the highest level of CXCL10 induction following HCV infection. And you might also notice that we had to normalize, because the cells were differentially permissive, we had to normalize these results based on the amount of viral RNA, because if the cell is replicating to HCV to different levels, there will be different levels of HCV RNA exposure. So we also did experiments using high MOI synchronous infections, where basically the same amount of viral RNA went into cells at the beginning and would trigger similar responses. And the bottom line is that we still found that the double positive cells produced the highest CXCL10 induction. And we also did experiments where we selectively knocked out TLR3 and RIG-I and found to confirm these responses. So for the rest of this portion of my talk, I'm going to be talking about these double positive cells, because the data clearly show that both TLR3 and RIG-I are required. So the next question we wanted to ask is, what is the role of type 1 and type 3 interferons in this process? And the reason for looking at interferon is that CXCL10 is an interferon-stimulated gene. It's induced by all types of interferons, interferon alpha, beta, type 2 interferons or interferon gamma, as well as type 3 interferons, the interleukin 28 and 29. And as I mentioned, CXCL10 is induced in all models of HCV infection, and that in interferon-stimulated genes are highly induced in the livers of interferon non-responsors. And recent studies from Barbara Rehrman's lab and Emmanuel Thomas have shown that type 3 interferons are major players in interferon-stimulated gene induction in human liver. So we basically asked the question, are interferons required for this? So the way we approached it is, first, well, let's just dump on interferon and see what happens. And because no one had really looked at CXCL10 induction in response to interferon in these cell lines. So you can see that interferon alpha caused a dose-dependent increase in CXCL10 mRNA, as did interferon beta. IL-28B was also able to induce CXCL10 mRNA, although we didn't really see a dose response, but I think we maybe started with too high of a concentration. And similarly, IL-29 induced a dose-dependent increase in CXCL10 mRNA. So basically all types of interferon induce CXCL10, and it's true in these cell lines, in this cell line. So to approach the interferon requirement question, we first did a kinetic experiment with the simple idea that if interferon is driving CXCL10 during HCV infection, you should see interferon production before CXCL10. And the bottom line is that we don't really see much interferon produced, and when it is produced, it's either at the same time or after interferon production. And this is consistent with sort of defective type 1 interferon responses that have also been observed in these cell lines, or in the Huh7 background. But to get at this a little bit more quantitatively and objectively, we decided to neutralize interferon in the culture and look at the effect on CXCL10 induction. So the way we did that is we used this really neat protein from vaccinia virus called B18R. And this is a soluble type 1 interferon receptor that is produced during vaccinia infection, and it mops up type 1 interferon that's released by cells. And just to show you that the system works, if you treat with interferon alpha and examine at 24 and 48 hours after treatment, you can get induction of CXCL10. It kind of goes away by 48 hours, but B18R completely neutralizes the interferon induction of CXCL10. So what happens when we look in the context of HCV infection? Well, you can see here that HCV, as before, you see this time-dependent increase in CXCL10, but absolutely no effect of B18R or interferon neutralization. And this is for CXCL10 mRNA. And if you look at the protein level, you see the same thing. Mock infected cells have baseline expression of CXCL10. Virus induces quite a large induction of CXCL10. B18R or interferon, type 1 interferon neutralization doesn't do anything. But again, the positive control interferon induces CXCL10, and that's neutralized by B18R. So these data indicate that type 1 interferons are not required for CXCL10 induction during HCV infection in this particular model system. And because of time limitations, I'm just going to tell you that we did the same thing. We did interferon, type 3 interferon kinetics, and we neutralized type 3 interferons with a pan-specific IL-28 and IL-29 antibody and found the same results. So we're pretty convinced that in the Huh7 model, there's no requirement for type 1 or type 3 interferons. And to really belabor the point and drive the point home, we decided to do some immunofluorescent analysis. And in the green is the expression of the HCV core protein, and the sort of orange is the CXCL10 detected by immunofluorescence. And if you just get the computer to draw a line through this region and you measure the CXCL10 and core levels, you can see that as core increases in this plane, the amount of CXCL10 protein also increases. So seems to be a direct relationship of HCV core protein expression and CXCL10 protein. And so to really get at this, we decided to compare the pattern of the immunofluorescent staining pattern of CXCL10 induced by an autocrine or a paracrine stimulus. So we simply treated with interferon gamma and TNF, which is a known inducer of CXCL10. And you can see that basically all cells express it, and it's pretty homogeneous. But if you look in the virus-infected cells, you tend to see more CXCL10 where there is viral protein expression. So like the collagen talk from earlier today, we actually established a training set where we taught the computer how to detect images that didn't express any HCV core or CXCL10, expressed one or the other, or expressed both. And with that training set, we had the computer then classify the level of expression, and we queried over 2,000 cells. And the results are shown here, and you can see that HCV-infected cells had significantly higher levels of CXCL10 signal intensity. And furthermore, the core signal really nicely correlated with the CXCL10 signal intensity. So the viral pattern of induction of CXCL10 does not look like a paracrine stimulus, and further supports our argument that it's not that interferon has no role, or any sort of secreted molecules have no role in CXCL10 induction. So the conclusion for this part is that RIG-I and TLR3 sense HCV infection and are required for maximal CXCL10 induction during virus infection, and that neither type 1 nor type 2 interferons are required for induction of the chemokine in these cell lines. And so you may say, well, that's fine and dandy, it's a hepatoma model. And if you haven't thought about this, you should be asking this question. We need to go into primary cells. So we did, and we asked, does HCV induce CXCL10 in these cells, and are interferons required? So the first thing we did is we looked at some various donor preps of primary hepatocyte preparations. And I should say that these were obtained either commercially from Invitrogen, or we got them through an NIH-sponsored tissue distribution system. So we've looked at about 8 or 9 different donor preps. But this just shows that in this one particular prep, you can see that both of these preparations express both RIG-I and TLR3. and that these two donors respond to a TLR3 trigger with poly-IC or RIG trigger by transfecting poly-IC, and they both induce CXCL10 mRNA, although there is, as you'd expect, donor-to-donor variation in the response. But this also correlates at the protein level. So these primary hepatocytes, they have RIG-I and TLR3. They do what we think they should do when they sense HCV or when they sense RNA. And so we repeated the kinetic studies that we did in the Huh7 cells in the primary hepatocytes. And what we did is we measured type 1 interferons on the top and type 3 on the bottom. So interferon alpha is in white, interferon beta is in the gray. CXCL10 is in the triangles, and we also measured accumulation of HCV RNA over time. So for type 1 interferons, we noticed minimal type 1 interferon production, but if we did find induction, it was at the same time as CXCL10. So again, if interferon was driving CXCL10, it should occur before, but we've never observed that. Similarly, in the type 3 interferon, we had considerably more robust responses, which is consistent with what Barbara Ehrman's lab has recently shown. But again, the timing of the type 3 interferon and CXCL10 was always coincident. The interferon never preceded the CXCL10. So then we, of course, the more formal experiment is to do the neutralization, and this is where we got really stumped because we were expecting no effect. If we neutralized interferon, there would be no effect, and we found actually the opposite. So I'll just take you through this. Here's HCV infecting primary hepatocytes, and you get a really good CXCL10 response, and you can see that at 24 and 48 hours. But we actually found that if you neutralize type 1 interferons with B18R, we really attenuated the response. And at this also, it was observable at the protein level. You see HCV inducing the protein, but B18R really neutralizes it almost completely at 24 hours, but there's some recovery. So we were really kind of shocked because we were going along this and thought, yeah, this is going to be a great story. And so we scratched our heads a little bit, and we thought that if the hepatocytes, we think, require no interferon, we actually started thinking that it's probably contaminating non-parenchymal cells in the primary hepatocyte preparations that are producing the interferon in these preps. So to get at this issue, we first wanted to demonstrate that non-parenchymal cells are contaminating primary human hepatocyte preparations. And for that, we used a Fluidigm real-time RT-PCR platform that we looked at 48 gene targets, including 21 cytokines and chemokines, including CXCL10 and some other immune cell-derived cytokines like IL-10. We also focused in on leukocyte lineage markers for dendritic cells, macrophages, and monocytes, and also 14 genes that are tend to subtype markers for macrophages, including the M1, M2, and Kupffer cell lineages, and then we had 2 reference genes. We looked at 9 primary hepatocyte experiments from 8 unique donors, and we had various treatments with JFH1 infection, Sendai virus, poly-IC, interferon treatment, IL-28B treatment, and we also included mock and infected the double positive Huh7 cells. But because of time's sake, I'm just going to show you the data from the HCV infection, because I think it's most relevant to the story that I'm trying to tell you today. So the bottom line is that so here are the lineage markers, the chemokines, the lineage markers, the macrophage markers. The scale over here is on the right. The red is highly induced, black is not detected, and sort of intermediate levels of induction are in between those. Across the top are the different primary hepatocyte preparations. These are 8 unique donors that were infected with HCV. And the first thing you can see is that CXCL10 was quite strongly induced in all of the infected hepatocyte preparations. There was some expression of interferons. We noticed some expression of interferon gamma. We also noticed some immune cell cytokines like IL-10 and IL-23. And then with the lineage-specific markers, we found variable expression levels of DC sign, which is a dendritic cell marker, and some monocyte expression of monocyte markers, although these were relatively low, as well as these CXCL10 CR1 monocyte lineage markers. And for the macrophage subtype markers, we saw a number of genes that were induced, including NOS2 and in particular, the Kupffer cell marker CD86 was fairly well represented in this prep. So we feel these are pretty compelling evidence that pretty much any primary hepatocyte preparation that you either buy or you obtain from an academic lab has non-parenchymal cells in it. So to figure out the role of these non-parenchymal cells and interferon on the CXCL10 story, we decided to attempt a negative selection of these primary hepatocyte cultures. So we got it. This is a commercial prep from Invitrogen. We took half of these liver cells and just didn't do anything to them. So that's sort of the standard or normal primary human hepatocyte preparation. The other half of the prep, we put over a magnetic column with these magnetic beads to select out leukocytes using anti-CD45, macrophages and Kupffer cells with anti-CD68, and we used anti-CD31 to try and get any liver sinusoidal endothelial cells. And so the cells that came off the column were, we effectively believe that they were depleted of non-parenchymal cells, so we called them depleted PHH preps. And we also went back to the cells that stuck to the column and analyzed them. And we went back to our Fluidigm platform, and here are some of the results. So the standard preparation of primary hepatocyte looks like this. You infect with HCV at 24 and 48 hours. You see CXCL10 induction, some low-level expression of interferon, and actually some production of interferon gamma and IL-10, which are known to be produced by immune cells. You also see expression of some DC dendritic cell markers, as well as these different macrophage markers, and in particular CD86, which is a Kupffer cell marker. On the other hand, when you look at the depleted cell population, what's clear is that you still get very good CXCL10 induction, but the expression of interferon gamma and IL-10 and the IL-23 are completely gone, as are the monocyte and Kupffer cell markers. And if you go back to the cells that stick to the column, which are presumably your non-parenchymal cells, this is what you find. So there are a few genes listed here because these were the only genes that we could detect by real-time RT-PCR, and they included CXCR3, which is known to be on immune cells, some interferons, CD14, which is known to be on macrophages. And this is, I should tell you that this is a CT, not a full change. But again, this is high-level expression, and these are sort of intermediate levels. But these are the only genes that we could detect on cells that stuck to the column. So we felt like we had there were other cells besides liver cells in our preps, and we were able to separate them. So then, of course, the next question is, well, how does that affect the CXCL10 induction when you neutralize interferon? So what I'm showing you here is the standard prep, which behaves as the other prep that I showed you. You infect with HCV, you get CXCL10 mRNA and protein, and it's pretty much completely neutralizable with B18R. We see, and if you neutralize type 3 interferons with this pan-specific antibody, you can see some reduction in CXCL10. And when you look at the protein levels, both type 1 and type 3 interferon levels reduce the CXCL10 induction. So in standard preps, if you neutralize interferon, you reduce the induction of this chemokine. But when you look at the depleted preparation, the first thing you notice, if you take away the non-parenchymal cells, which contribute interferon, is that you see higher CXCL10 induction. And there's really no effect of neutralizing type 1 interferons or type 3 interferons. And that's true at the RNA level and at the protein level. So this means that if you remove non-parenchymal cells from these preparations, you no longer require type 1 or type 3 interferons for CXCL10 induction. So they look exactly like the Huh7 model, where, in other words, if you have a hepatocyte monoculture, You don't need any interferon. The hepatocyte is completely fine in making CXCL10. So if it's not interferon, then it must be over here. There must be some transcription factors that are directly activating CXCL10. And we figured that the two likely candidates would be NF-kappa B, and there's several sites annotated in the CXCL10 promoter. There are several binding sites. There are several sites for STAT binding and possible other interferon regulatory factors. So this part of the talk is that hepatocyte-derived interferons are not involved in CXCL10 induction during acute HCV infection. And the induction requires both TLR3 and RIG-I sensing, which leads to NF-kappa B and IRF3 activation and leads to direct transcriptional activation of the CXCL10 promoter. However, non-parenchymal cell derived interferons are involved in CXCL10 induction during acute HCV infection, and I believe they serve to amplify the intrinsic or endogenous CXCL10 induction in hepatocytes. So we're back to this model again, and so I hope I've convinced you that this side of the model, the hepatocyte itself, doesn't need any signals from over here to induce CXCL10. When these cells get to the liver, either from the periphery or from within the liver, by migrating to the site of infection, they can then contribute type 1 and type 3 interferons to further amplify this response. So I've really gone quickly, so I'd be happy to take questions. But I really want to thank Jessica Brownell. This is Jess. She's really takes costume parties very seriously. So at our annual retreats, she's gone as Carmen Sandiego, Uhura, I think here. She's also gone as a video game character. And you might think that people who work in my lab first names have to begin with J. So we've tried to include Erica into this by kind of renaming her Jerica. I've had some great collaborators. This work was primarily funded by a U19 application, which Hugo Rosen at UC Denver is the overall PI. And of course, I'd like to thank the conference organizers for giving me this opportunity to come up here. I really want to thank you all for the lovely views. It's just really incredible, very picturesque. And finally, I'd like to thank the band. So thank you very much.